Good morning, everyone. I'm Tanu, Soni, and I'm going to talk about, just continue with lipidomics and talk a little bit more about the data analysis and the report that we give to clients. So lipids, as Chuck and TM already introduced, can be considered as a subset of metabolome. And lipids are very important, just like any other omic profiling. Lipid profiling adds more knowledge to the understanding of the biological system. So my objective of my talk is going to be in 4 parts. I'm going to talk about lipid classes covered by our platform, and a little bit about databases and the nomenclature, because just to get it going with the data report that we give. And then I'm going to talk about the workflow of the platform, data processing and the data analysis. And then I'm going to give some example data sets. So for lipid classes, 3 main classes are covered by our platform: glycerolipids, glycerophospholipids, and sphingolipids. I think TM covered mostly in detail all of this. And this plot shows the class-wise distribution in the pooled plasma. So this pooled plasma is the primary control that we run pooled plasma in all our experiments. And it just gives the overview of the percentage of lipid in each of the classes. As you can see, PC is the most abundant one, and we see it in both modes, more so in negative mode, and triglyceraldehydes are also abundant ones. So databases, I want to touch a little bit about databases because of the reason, because a lot of times I keep getting these questions from people once we give the report. And I want to point out that, so first let's talk about lipid map structure database. So lipid maps structure database, when this lipid map project started, it really provided a great advancements for lipidomics. So this was the first internationally accepted classification system for lipid classes. And this is a great resource to find all the information about lipids. There are some tools that you can go and check out this website. And even though this is a great resource, I just want to point out that the report that we generate and the nomenclature, sometimes, when people come back, it doesn't make exactly sense to them. They ask more questions about it. So the lipid maps classification system, the way it works, and the lipids are classified. So you have a category, and then the main class, subclass, and then you have this unique identification, which is basically a geometric isomer. So this figure is from a paper from Lipid Home database, which is recent, I think, in 2013. And so they have added a little more classification knowledge to, they have just taken same lipid map classes and just added a little more information here. So in our platform, when we give you the report, we give you the report at this level, at the species level. Fatty acid scan species, subspecies, isomer. We can probably get to fatty acid scan species for a few lipids, and right now, and I think as time progresses and we advance this more, probably we'll get to most of the species. But this is what you get right now. So a lot of time people come back and ask, okay, this is PC36-2, but what is the fatty acid information? And lipid maps, if you try to search these things by directly by the names that we give, you won't get any information. So just a little about that. Okay, so this is a workflow very similar to all other metabolomic and targeted workflow. And it starts with when we get samples from the clients, we get all the information about the experiment design, and then we use it information to randomize. If blocking is required, we block the samples, and then we run it through mass spec. After that, we get information, we get raw mass spec data files. We run them through Lipid Blast, and then we get identification. So we run each of these sample files through Lipid Blast. So for each of these sample files, we get all the lipid hits. And then we combine the identification from all these samples and create a master list. So it's like creating a unique library for each experiment set. So once we get this library, which is unique for this experiment, then we sort of go in a targeted way. We take these identifications from Lipid Blast, we extract the identification, the mass, and the retention time, and then we quantify this on MS1 spectrum, again using the multi-quant software. And then that's how we get the peak areas. So two important steps: MS identification, MS based identification, and MS based quantification. So for MS based identification, we use Lipid Blast, which is the in silico database for MS spectra. It contains more than 100,000 compounds. A lot of them are, I don't think, really exist right now, but this is in silico. And it covers almost 26 lipid classes. So, so this is just an example to show how this works. PC, this is our internal standard, PC340 that we use, we spike in our samples. And this comes from Lipid Blast, and this comes from the experimental sample. And as TM already spoke about this, for PC340, for this class, this particular class, 184 is the major characteristic fragment that we see, and Lipid Blast matches these characteristics fragments and other fragments to give a score. Sometimes, and we select the topmost hit based on the score that we get from this identification. A little bit more information you get from this is basically all the other fragment information and and the formula information that what these each individual fragments can be from Lipid Blast. One more example is PE34. This is another internal standard that we use, and for PE characteristic neutral loss of 141, which is this peak 720 minus 141. This is the major ion that we see for the identification. So for quantification, once we get our library, we use all our samples and run each of these samples through all the identifications. And just like all other like metabolomics and targeted metabolomics, we get all the quantified peak areas. So for data, once we get this raw data as peak area, for the data analysis workflow is we do QA/QC on this data, and then we normalize this data, and then we do the pre-treatment, scaling, transformation, and then we go for univariate and multivariate analysis. QA/QC, three important things. Internal standards that we spike like a pool of all the internal standards for most of our classes we have internal standards, approximately 13 different internal standards we have, and pooled plasma as a primary control. This is basically that we do for all our experiments. We run these samples, and this gives us a good idea about our instrument performance also over a period of time, that how things reproducibility, how things are reproducible. Then test pool sample basically is the pool extracted from the that particular experiment that we get, and this also we use for QA quality assessment. So again, we have to think about the variations before we even go for analysis. So two kinds of variations. Some variations are coming from the type of sample that we get. So we need to think about this beforehand, before even we start this running these samples. We get tissues with different weights, and need to normalize this. So maybe additional analysis required, even protein content, or for normalization we use tissue weights for normalization. Urine creatinine level, or for cell lines, cell counts. So all these things for the variation we need to keep in mind. Analytical variance that happens due to sample prep, instrument drifts, or run order, batches. Internal standard takes care of mostly the sample prep, and if there is a drift in instrument, run order or something, then we use these quality control replicated measurements, which we use to correct for any batch variations, or if that we are not able to correct with internal standard. So internal standards, this is a plot from a QC study that we did for our stability of our platform. We ran, I guess, 3 batches of pooled plasma for multiple days, and we checked the reproducibility for the pooled plasma. We had, there was a lot of other data, other things that we did, but this is a good plot to show, because this internal standards are fairly reproducible, and as you can see, the variation within the day and between the days also is very low, RSD below 9%, which shows really good reproducibility. So this plot is from a paper which was about one of the normalization methods using multiple internal standards, and I just wanted to show this to just bring the context of whatever normalization we are doing. We need to keep in mind the type of variations that can happen with the normalization. Very often people use TIC based normalization, and as this was pointed out in this paper, it can create bias in the data. Sometimes it's just a pointer to keep in mind. Usually metabolites, they don't have, they're not, so basically this, when we do TIC normalization, we take into account that metabolites have the self-averaging property. What that means is that increase in one means there is going to be a decrease in one, which is not true. They don't work like that always. So if you normalize this by TIC, then you'll see a decreased in content of the phospholipids in this type of mouse, which is what was pointed out in this paper, was not the case, actual case. This is not low as compared to this one. So normalization methods, a lot of literature on this, too many methods. What works out best for at least lipidomics platform, in my experience, is if the internal standards are really good, just the normalization relative response to the internal standards works really well for normalization if it is required. Still, batch effects is something that we are not able to correct with the internal standard most of the time. So this method, Loess, which is local regression smoothing, is the method from this paper. This works fairly well. So a little bit about Loess smoothing. The way it works is basically the smooth value are determined by fitting the localized, fitting models to the localized area, and we use QC samples to get the degree of smoothing for all these samples. The QC samples are replicate measurements, and so we use the QC samples to train this and get the degree of smoothing that we can apply to these samples, and then we apply it to the rest of the samples. So one example from example data for the batch effects, we got 156 samples from client, and then no big factors as such. Given by the clients was only age and glucose level, all continuous, and we ran this data 156 sample in 3 batches and over a period of 7 days. So first we start with the internal standard. We check the quality of our internal standards, and most of the internal standard work pretty well except the TG510. So once we check this, we see this is not working out well. We throw this one, and then we use the rest of the internal standards for normalization. Checking the variations in our QC samples, our primary and secondary controls. As you can see, the pooled and test pools are really low. This is basically the RST on the y-axis. So as you can see, pooled and test pools have really low RSDs as compared to our actual samples, which is what we would expect for the quality of the data to be good. So in this data set, we see big batch effects between the 3 batches, all 3 batches shown with different colors. We normalized this data to internal standard, and we could still see some batch effects with this, especially batch 1, which is red here, was not able to correct for all the batch effects. Then we used Loess, the method that I just said, and we were able to correct all the batch effects. Okay, this second example I want to show as an example for the report, that once you submit the data, submit the samples, what kind of report you can expect from the core. So we got this nerve tissues in 48 for this mouse model, and basically this was the mouse model and the study. They were interested in the nerve disease, and 6 different types of mouse, and within each category they had 4 samples for each high fat and control diet fed mouse. So we get this information, this much information from people, and we use this information first to randomize and run the samples. This is the TIC for all the samples, and it's fairly good for a tissue data set. You do see some variations, but as I said, they are different tissue weights also, and tissue is not as clean as plasma is always. So this is just basically, once we get this, first thing with all the raw data, just do a PCA plot to see how all the data is, our QC samples are looking here. As you can see, they are clustered really well together here. There's no obvious grouping in the raw data right now, the control and high fat diet. Then we look at the internal standard for positive mode. All of them work really well. So we use all of them for normalization. Negative mode internal standards look pretty decent, all of them below 25%. So again, we use all the internal standards for normalization. Once we get this QC check, we use weight. In this experiment, we use tissue weight to normalize, tissue weight and internal standard to normalize the data. And then we get same, you don't see any obvious patterns in this data. So sample report consists of basically an Excel spreadsheet with all the sample information, and I'm just going to walk you through each individual tab. Along with this, we give a document, basically a document describing all the methods that we apply, the procedure for the data analysis, and also method for sample preparation and all other information. Basically, whatever we do with the data, we give you in a written form also. So this Excel spreadsheet, first tab, basically the sample information, what we give internal IDs to all your samples and what corresponds to what all the metadata that we get from you, we give together in this. Then the raw data. So for a lipidomics report, the raw data, you'll get the name of the compound, and this is the RSD for each of the compound, basically from the test pool samples, which are secondary control, to give you a quality assessment of how good that particular compound, particular lipid is. Then the class, what class they belong to. Then the start mass and end mass gives you the window that we used for the mass range window that we use for identification, and the retention time, and the tag known, unknown. So this, we decided to give this as known, unknown based on all the lipids that we give are named, but we see about 10 to 15% of odd chain fatty acid lipids, which is not so usual to see in plasma or even in any tissue samples, except I think bacteria. But we see about 10 to 15%. So we usually give a tag as unknown to the odd chain fatty acid lipids, and this can be like a false identification. So just to give an idea that to keep in mind. And of course, the raw peak areas for each of the values. Then we give you the normalized data set for that particular experiment, both positive and negative mode. We give separate raw data and normalized data. Then a simple basic analysis. If you have defined groups, we usually give you a basic t-test or ANOVA, univariate analysis, mean values in each of these groups. And again, using this, only the significant lipids from the t-test, we did the PCA again, and we were able to separate these high fat and control fed samples clearly from each other. And then we give whatever information with this PCA, all the loading information, we give both in positive and negative mode. This is another plot for each of the mouse types, that additional factor that we had here. Again, too many groups, just 4 samples, couldn't differentiate all of them, but still a good separation between the high fat and control mouse. So this is the extent of the report, basic report that we give for to the clients with the preliminary data analysis. So this third example is just another data set example, and this is something that we are working internally. TM is working with prostate cancer. We talked about another example, and this basically the objective is to delineate the lipidomic alteration that occur in prostate cancer progression, and we used cell lines, prostate-derived cell lines in this experiment. Benign RWP is a benign prostate cell line, and then we used 4 different prostate cancer cell lines: VCAP, LINCAP, DU145, PC3. We identify more lipids actually as compared to in cell lines as compared to other biospecimens, and we got almost half of them significant. And this is just a PCA plot to show the all these. It's not very clear, but all these cluster really well in PCA. This is the benign one, and all the cancer are really far from this, except this one. This one is also DU145 is different from rest of them, how they cluster here together. So the most significant differential lipids, all these lipids, phospholipid metabolism, this makes us conclude phospholipid metabolism increases during cancer progression. This is a heat map with for the z-scores of those 20 most significant lipids, and this hierarchical clustering shows proper separation of each of these of our groups here. And as you can see from this is our benign, the black ones are the benign ones, and you can see all the changes in the level in this. Red is high and green is low, and they are not very clear, but they are the same lipids that I showed you before. Again, one more thing we did in this is to distinguish the androgen-dependent cell lines from androgen-independent cell lines. So VCAP, LINCAP are androgen-dependent, and PC3, DU145 are androgen-independent. So we did a differential analysis, like a t-test in between these two, and identified 415 differential ones. And the most significant, this is the list of the top ones, and you can see the again phospholipid and glycerolipid metabolic lipids. All these are. This again shows how different, totally opposite direction changes in these two androgen dependent and androgen independent. So this is still, we are working on this for further experiment with the biology and all, but just wanted to show you the basic analysis that we've done. Any questions?
Is your quantification based on absolute intensity of peaks, or based on the ratio of sample peaks to internal standard peaks?
Yeah, it's the ratio. It's not absolute quantification. We don't do absolute quantification for any of the untargeted. This is the untargeted form.
So if it's a ratio, you have limited number of internal standard. So when you do the ratio, how do you pick the different lipids at different internal standards?
You mean for normalization?
I mean, you have 1,000 different lipids.
Okay.
But you don't have, let's say, 10 internal standards.
Yeah. So basically, for lipids, you pick it class-wise. If we don't have a lipid for class-wise, you can use the median of all other, or you can use the one that you think is most similar in the retention time. So there are different approaches to do it. We have 10, which covers almost all classes except I think 1 or 2. Does that answer your question?
Oh, yes. So you are saying mainly based on retention time, or based on the
Basically based
similarity
on,
of the
yeah,
chemical
so
structure?
so the idea, especially in lipidomics, is the similarity in structure is physical or chemical properties we define by class, right? That's what we is different from metabolomics, that we have class information in lipids. So we can use that class information and classes, obviously retention time also. Each class has a different retention time. Basically, some classes share retention time, but they elute at different retention times. So class based or retention time based, mostly class based here in lipidomics.
Okay, so did you do any sort of cross validation? I mean, you use different interstandards for the same lipid. Will you get the same results, or will it be very different?
So if you use
Let's say you are measuring lipid A, you have 10 choices in terms of internal standards.
yeah.
So have you seen any difference when you use different internal standards for the quantification?
Yes, you can if you use, it depends on like when you're using this, basically you would use class-based internal standard. But what you're saying, yes, if you use different internal standard, there is a possibility that you will get different results. And sometimes, I've seen that some internal standards, when they don't work so well, and like I said, like let's say TG doesn't work really well. So either we use a median of all other internal standard, or I can pick the closest one, that is CE cholesterol ester appears in the same retention time, to choose for a normalization. And that just really depends how it's changing your data. If the way I usually do is I check the variation. If it's increasing the variation in my test pool too much when I normalize it, I don't use that internal standard. I use some other method. I use median or something else. If it's decreasing the variation in my RSD samples, for RSD for my test pools, I feel that it's working better. It's working pretty well.